Someone has, let's say, cancer. You can get a blood test done, and based on the results of the analysis, the doctor can first make a diagnosis, and second, based on the results, the doctor can make decisions about the best medication that you can take. You may respond to the treatment, or you may not, but at least we have the tools to make a diagnosis. In psychiatry, that is totally different. We don't have any biomedical tool that we can use to make a diagnosis and to take decisions about the treatment. We have interviews with the patient, and based on how the patient behaves during the interview, we decide about the potential treatment. For example, an antidepressant. They may respond, or they may not, but we don't have the measures to make decisions about the best medication that this individual may take. And this is one of the reasons why, in my laboratory, we are interested in investigating at the molecular level how medications used to treat psychiatric conditions work. Until recently, when we heard the word psychedelic, what we thought is about a psychoactive drug that induces hallucinations, or changes in what you see, you hear, or you smell, and you think, you feel that they are real, but they are just part of your mind. Because of that, psychedelics have been used recreationally and for spiritual purposes since the beginning of human history. And one example is, for example, psilocybin, the active compound in the hallucinogenic or magic mushrooms. But while these drugs do induce hallucinations, recently we are trying to understand or to investigate if they can also be used therapeutically in individuals with, for example, depression or other psychiatric conditions. And as an example, patients with depression that don't respond to any medication, they are what we call treatment resistant. With one single administration of psilocybin, they saw benefits. Not only that, these benefits are long-lasting for weeks or even months after one single administration of this psychedelic. How can that be? How only one administration of psilocybin can induce such profound effects in individuals with depression, anxiety, and other psychiatric conditions. Receptors are proteins located at the cell surface. They recognize and bind to signal molecules. For example, what we have in the back of our eyes, in the retina, is called rhodopsin, and is a photoreceptor that detects light. Using different molecular and biochemical tools, we were able to identify a neuroreceptor that is responsible for all the hallucinogenic effects of psychedelics, and it is called the serotonin 2A receptor. Those experiments were done in animal models, in mice, and that finding was replicated by other groups using pharmacological tools to block this receptor. And they found that the hallucinogenic effects of psychedelics were prevented. That is called in pharmacology an antagonist. So yes, now we know that the hallucinogenic properties of psychedelics are mediated mostly via this serotonin 2A receptor. But how about what I mentioned before? How about these potentially positive effects of psychedelics? And one of the mechanisms is what we know as plasticity. In neuroscience, plasticity is how the neurons can change structure, function, and connectivity in response to new information. And this new information can be, for example, sensory information, can be development, or can be, for example, pathological reasons. In the brain, plasticity is how the neurons get rewired in response to new information. And in the laboratory, we have different tools to test this neuroplasticity. For example, we can measure synaptic plasticity or long-term potentiation and how the neurons connect differently. We can measure structural plasticity. They have like small spines that they use to connect with each other. And we can also measure epigenetic plasticity, or mechanisms, molecular mechanisms that control gene expression in different cells. And we're aware that in individuals with psychiatric conditions, they have alterations in these neuroplastic processes. And what we know is also that a single administration of psychedelics can also induce profound effects in these synaptic plasticity processes. They affect functional plasticity, they affect the structural plasticity, and they also affect epigenetically gene expression. So that is very clear now. But because these results, even when they are promising, they are also relatively new, we still have many questions that are still open. For example, do you need to induce hallucinations to have these beneficial effects in terms of synaptic plasticity? Could it be possible from a therapeutic perspective? Could it be possible to design new medications that are able to affect plasticity but do not induce hallucinations? And in a different way, in patients, do you need the trip to see the clinical effects of psychedelics? Do you need this mystical experience for them to see benefits in terms of depression, anxiety, or addiction? In science, when we do experiments, we always need to include a control in the experimental design. And in clinical studies, the control is what is called placebo. And this is a group of individuals that think they're receiving the active molecule, but actually they're receiving something totally neutral, for example, a pill of sugar. But think about this for a second. In clinical studies testing the effects of psychedelics, it is very obvious for them to find out if they are receiving psilocybin or placebo. So this opens a very intriguing question if these therapeutic benefits of psychedelics is caused by themselves or by something else like the placebo effect. Putting together collaborations among different disciplines and different groups, combining basic, translational, and clinical research might find a way to find a solution to all these still open questions. Thank you.